All right. Hello, everyone, and thank you for joining us today for this iRepertoire webinar. I'm Jenna Gallegos from Samba Scientific, and I'll be the moderator today. In this webinar, Dr. Kristen Balog and Dr. Asaf Paran from BioNTech will present data on their study of peripheral blood cells from metastatic melanoma patients before, during, and after treatment with personalized neoantigen therapy plus anti-PD-1. So without further delay, I'll turn the time over to our speakers, Kristen and Asaf.
Thank you very much, Jenna, for the introduction. Again, my name is Kristen, and my colleague Asaf and I will be presenting today on utilizing TCR repertoire sequencing to predict response to immunotherapy in melanoma patients. Next slide. So the work that we'll be presenting today is actually a publication that came out of our group last year in Cell Reports Medicine, and that publication is shown here in case anyone wants to follow up in more detail on the study. So before I pass it over to Asaf to get into the data, I want to give a little bit of background on the personalized neoantigen vaccine trial platform that we're going to be talking about today. So this is NeoPVO1, as I mentioned, a personalized neoantigen vaccine platform. And the reason that we sort of got into this platform is that we know that neoantigens are really important for driving an effective anti-tumor immune response. But most patients aren't able to mount a broad neoantigen-specific immune response on their own. We also know that in the clinic, most patients don't respond fully to checkpoint therapy. So we developed this platform to focus a patient's immune response on those relevant tumor neoantigens that can hopefully lead to improved clinical outcomes. And this involves a custom designed set of manufactured peptides for each individual patient's tumor mutations. And the schematic on the right outlines sort of how we go about generating these peptides for each individual patient's tumor. So a patient comes into the study, we take a biopsy of their primary tumor, and we perform next-gen sequencing on that to determine the neoantigen profile of that patient's tumor. That information then is fed into a proprietary bioinformatic pipeline that we've developed in-house called Recon, which essentially predicts which of those neoantigens are going to be processed and presented by MHC. And we're given a ranking for those peptides, of which we pick the most likely to be processed and presented. And then we manufacture those personal peptides at a GMP level, and they're given to the patient intramuscularly throughout the vaccine time course. So the specific trial within this NeoPVO1 platform that we'll be talking about today is called NT001, and we'll be focusing on a cohort of melanoma patients within that trial. So on this slide, I want to focus on the 3 time points that are shown in the schematic. So we're able to draw leukapheresis blood draws at 3 different time points listed here. The first of which is the pre-treatment time point, and that's taken at week 0. And this is essentially before the patient is dosed throughout the therapy. And then we take a second time point at the pre-vaccine time point shown here at week 10. And this is taken after 10 weeks of nivolumab monotherapy. And all of these patients are put on nivolumab monotherapy during the time where that peptide vaccine is being designed and manufactured. And then the third time point is the post-vaccine time point, and that's drawn at week 20. And this is after the patients have been given their first 5 priming doses of vaccine, as well as the first booster. And they're then given a second booster dose after that time point. And we follow these patients out to 2 years on study. And then what we're going to be showing today is from these blood draws, we're able to perform TCR sequencing, as well as detailed immunophenotyping by flow cytometry. And there's more detail on this study in the publication shown on the bottom right from Ott et al., which also came out of our group last year. And this goes into a lot more detail on the clinical outcomes, as well as the overall immune response analysis to the vaccine itself. So the last bit of background information that I wanted to give everyone today is a little bit of the overall clinical trial results and how we're bucketing these patients as responders or non-responders. So you can see in the waterfall plot on the top left, as well as the swimmers plot on the bottom right, or sorry, the spider plot on the bottom right, that we can bucket these patients based on RECIST criteria into 4 categories. So we see in red that we have a number of patients who experience progressive disease, as well as patients in gray who had stable disease. Blue is a partial response, and then we had 1 complete response shown in green. So given this information, we actually decided to bucket these patients as responders or non-responders based on whether or not they achieved progression-free survival at the 9-month time point. So a patient from this point forward in the talk that's called quote unquote a PFS9 patient is considered a responder. And that's a patient who didn't experience progressive disease by 9 months. And we had 14 patients that fell into this category. Conversely, the no PFS9 patients are the non-responders. And these are patients who did experience progressive disease by the 9-month time point. And we have 7 patients that fall into this category. So I think, these sort of outcomes are relatively common for patients undergoing immunotherapy in the setting of melanoma, where we see a lot of patients that do respond to some degree to this treatment modality, but there are a number of patients who just don't respond and progress relatively quickly on treatment. So what we wanted to understand in this study is there anything that we can observe sort of in terms of immune phenotypes in these patients, either before or during treatment, that correlates with whether or not they respond to the treatment and can potentially predict whether or not a patient is going to respond to this treatment modality. So from here, I'm going to pass it over to Asaf, and he's going to start to answer that question.
Thanks, Kristen, and hi, everyone. So in order to answer that question, we first need to be familiar with some concepts regarding T-cell receptors and their repertoire, which we've used in this study. So the T-cell receptor is a molecule which recognizes antigens that are presented by other cells, and it doesn't have a fixed sequence. It's not just one receptor. In fact, there's a huge diversity of potential molecule, and there are different estimates out there, but it's really an enormous number. And this diversity is achieved through 3 mechanisms. So first, just to mention, the TCR receptor is composed of two chains, the alpha and the beta, that are generated through random rearrangement of segments called the V, the D in the case of the beta chain, and the J segments. And for each of these segments, there are multiple copies in the genome that are slightly different from one another. So when these pieces get recombined together into one segment for each of these 2 chains, we are getting a very significant combinatorial diversity from which V was chosen from the pool, which D, and then which J. But on top of that combinatorial diversity, probably the most diversifying factor to the number of potential sequences is the fact that when these segments are brought together, the V and the D, and then the D and the J, or directly the V and the J in case of the alpha, there is a process of nucleotide deletion or insertion that is non-templated at the junctions between them. So these sequences were not originally encoded in the genome, and that is obviously creating a very, very wide sequence space of possibilities for TCRs. And then we need to remember an alpha can pair with it. It should pair with a beta chain, and that is also contributing to the diversity of the repertoire. So when we think about repertoires, it's basically the collection of all the TCRs that we can get in a sample. In this study, as Kristen has already introduced, we've collected the repertoires of TCRs from peripheral blood in our patients. So when we think about this process of VDJ recombination, where the T cells basically receive their TCR identity, we're looking across all the different cells. And upon antigen exposure, what we expect to happen is that the cell population that is actually recognizing that particular antigen will undergo clonal expansion. And when we think about it from the perspective of a repertoire, when we get the data from the repertoire sequencing, each TCR, or in essence, each T cell clone, gets a count. So it's a weighted set of measurements that tell us some TCRs occur more than others. And we can quantify that as a frequency. So what fraction of the repertoire is occupied by a particular clone? In this scenario that is completely made up, of course, we're demonstrating a clonal repertoire. So there's a clone that's taken a significant portion of the repertoire's frequency. In contrast, we can imagine a scenario when there's no antigen exposure. And in that case, the different T cell clones do not undergo clonal expansion, and we expect the TCR counts to be relatively similar to one another. This is a more maybe diverse repertoire, or at least in our definition, less clonal, and the frequencies should all be relatively low, and there will be many of them. So in our study, what we wanted to do first is to look at the repertoire sequencing that we got from our patients and bucket each clone into a different category by its size. So rare clones were less frequent than 1 in a million, and small and medium and large and hyperexpanded are according to these frequency definitions. And then we can look across our PFS9 patients, which, just as a reminder, this is the responders groups, a group of 11 healthy donors that we also incorporated into this study, and the no PFS9 group. And what we're seeing is that even from this very big picture view, we are already seeing shifts in the different categories between PFS9 patients and both healthy donors and no PFS9 patients. And those shifts, you can maybe start seeing them in the sizes of the rare and the medium, the small and the medium, but especially when we focus on large and hyper-expanded clones and how frequent they are in the repertoire, we see that PFS9 patients has a significantly larger fraction of their clones being large, as well as having hyper-expanded clones, which are barely, if at all, detected in healthy donor and no PFS9 patients, so the non-responders. And we can delve deeply. So this view on the left, it's just an average across all the samples, all the patients by their response state, but regardless of time. So now if we break it down by what we learned from pre-treatment, pre-vaccine and post-vaccine time points for the large clone category or the hyper-expanded clone category, you will see that especially for pre-treatment and pre-vaccine, we see a significantly higher fraction of clones that are large, that are in the large size buckets, comparing with both healthy donors and the no PFS9 patients. For hyper-expanded, we see that same results occurring in all 3 of the time points. So we wanted to try to take a bigger picture view of the repertoire, because assigning the clones into buckets is sometimes arbitrary. How do you decide what is a cutoff for each of the buckets? So to summarize the clonality of the TCR repertoire, we use a mathematical coefficient that's called the Gini coefficient. And this is a coefficient that's borrowed from the field of economics, and it tries to estimate the distribution of wealth in the population when applied to economics. It ranges between 0 and 1, where 0 means wealth is evenly distributed, and 1 indicates all the wealth is held by one person. And in this diagram that we've brought from the internet to demonstrate the concept, we can look at the cumulative share of observations. So let's say the different persons in the population and how much in aggregate they contribute to the cumulative share of the values or the wealth in this case. So the closer the curve is, the purple curve here goes to the bottom right. That means that more people contribute less to the total wealth, and fewer people contribute the rest of the wealth. So that is demonstrating to us that Gini goes higher as this goes to the corner here. In the concept of TCR repertoires, we think about the scenario where Gini is closer to 1, where one clone is actually dominating the repertoire. So it has a very high frequency in the repertoire. And a Gini of 0 would indicate that all clones are exactly of the same size. So the TCR counts are equal for all. So when we apply it to our samples, and again, the same categories, PFS9 for responders, healthy donors, and no PFS9 for no responders, at the 3 time points, pre-treatment, pre-vaccine, and post-vaccine, we see there's a significant increase in the Gini coefficient of the repertoires of the PFS9 patients compared with both healthy donors and the non-responder population in all 3 of the time points. Another common coefficient that's used in the field to estimate clonality is the DE50. It's asking a different question, but it's trying to get at a similar concept. How many of the biggest clones cover half of the repertoire? So in this made-up example, we're saying, let's say there are 100 clones in a repertoire, and the total counts of all of these clones is 20,000. So we sort the clones by their size from largest to smallest, and we say the first one contributes 5,000 to the count that covers a quarter of the repertoire. Then we add a second one, we're now at 8 over 20, that's 40%. And we keep adding clones until we hit 50 on the repertoire coverage. In this case, this is clone number 3. So 3 clones out of 100 covers half the repertoire. That means that the D score for this imaginary scenario is 0.03. So when we apply that data, this approach, to our data, what we see again, PFS9 patients have a significantly lower DE50 score comparing with healthy donors and no PFS9 patients. And that is true for all 3 time points. So whether we're looking at the increased Gini or a decreased DE50, both of these measurements indicate an increase in clonality in our responder patients. And it is important for us to mention that we checked and we wanted to see whether this clonality is driven by viral antigens. So we're performing an ELISpot assay where the cells are introduced to a pool of common epitopes from across different viruses. And we see no significant difference between the responses in PFS9 patients and no PFS9 patients to these viral antigens, suggesting that the clonal expansion is not virally driven. So to recap the data that we've shown until now, the TCR repertoires of PFS9 patients, again, the responders, are more clonally expanded than either patients with no PFS9 or when we're comparing them to healthy individuals. And this increase in clonality is persistent from the pre-treatment and throughout the treatment. And that really brought us to our next big question. How are these large clones, are these large clones the same clones throughout time? Or do these patients have these big clones but they change from time point to time point? So the first thing we did is to apply another coefficient, and you'll excuse me for introducing all of these, but this is called the Jensen-Shannon divergence. It's another mathematical operator. Not going to go into the details of how it works, but it's basically allowing us to take 2 repertoires and compare them to one another. And the value that it gives us is basically a divergence score. So when the value is high, that means that the repertoires are divergent, meaning they don't have a lot of overlap. When we're using this metric across time points, it's basically giving us a proxy for the stability of the repertoire. So are clones similar between an early time point and a later time point? So first we applied this metric to our PFS9 patient samples and no PFS9 patient samples when we're comparing the pre-treatment, so the first sample, to the pre-vaccine, which is the second sample. And we see a significantly lower score in the Jensen-Shannon divergence for the no PFS9 patients. And remember, the scores for divergence, so low score, is actually indicative of stability. The same result was derived when applied to the pre-treatment sample versus the post-vaccine samples within each of the patients. So that is indicative that the PFS9 patients have more stable repertoires, but the Jensen-Shannon divergence has a limitation, and that's the pairwise aspect to it. So we wanted to apply a more holistic approach, which we chose the Venn diagram thinking to this problem, indicating that each of our 3 samples, the pre-treatment, the pre-vaccine, and the post-vaccine, is a circle in the diagram. And the different regions indicate how many times any clone was found. So if, let's say, a clone has only been found in the pre-treatment sample, it will go to area A. If it's been found in both pre-vaccine and post-vaccine, but not pre-treatment, it will be in F. And G indicates the clones that have been found in all 3 of the samples. And one thing that's important to note here as we look at the results from this type of analysis is that these regions don't count how many unique clones are in each region. It's actually weighted by their frequency. So when we move forward to look at the sizes of this different regions, so the size of A or the size of G, we will sum over the frequencies of the clones in that particular segment. And here's the result. So when we're looking at the G region, which again, that's the region representing clones shared by all 3 time points, and we take their cumulative frequency, we see that in all 3 time points, pre-treatment, pre-vaccine, and post-vaccine, the cumulative frequency of the persistent clones is significantly higher in PFS9 patients. And this increase in persistent clones comes at the expense of the not persistent clones. So we'll see that for regions A, B, and C, which is the regions indicating a clone has only been found once, there is a reduction in their total cumulative frequency there. And one visual way to think about this is when we're thinking about the PFS9 patients, they will have a more tight or a more significantly overlapping set of circles because their G region is higher at the expense of smaller A, B, and C regions, and vice versa for the no PFS patients. And as I've introduced when we were discussing what exactly is the Venn diagram representing. Until now, we've calculated the cumulative frequency, but we did have a question. So is the G size increased in responders because they have larger clones, or because they have more clones that are persistent, or a combination of both? But when we count how many unique clones actually are found in the G segment of PFS9 patient and no PFS9 patient, there's no significant difference that was detected between the groups. So this is suggestive that the cumulative frequency is driven by the respective size of the clones in these segments, not by the number of clones in these segments. So we'll quickly recap what we've learned about the stability of the repertoire. So the TCR repertoires of PFS9 patients, the responders, have less turnover from one time point to the next. And the clones that are found in all three time points in PFS9 patients are more clonally expanded. And that led us to the natural question, what is the connection between clonal expansion and repertoire stability? And I'll hand it off to Kristen to tell you more about how we address this question.
Thanks, Asaf. So as he alluded to on the previous slide, we're interested in correlating the TCR clonality and stability. So essentially that's what this plot here is showing, where we're looking at the Gini coefficient on the y-axis, which is a measure of the clonality, and then that cumulative frequency of consistent clones, or population G, that's shared between all three time points on the x-axis. And when we correlate these two, we see a nice positive correlation between clonality and stability. And we also see that the PFS9, or the responder patients in green, are sort of clustering in the upper right-hand corner of the plot, whereas the non-responders are clustering in the lower left, suggesting that the patients that are responding are showing increased clonality and increased stability of that clonality over time. So this sort of gave us the idea that there are possibly 4 states to the TCR repertoire that can exist along these axes of stability and clonality. And this graphic here outlines those 4 potential states. So if we say look in the top left quadrant to start, where there's high clonality but low stability, we can see using the size of the pie slices to represent the frequency of an individual clone, so a larger slice representing a more frequent clone, and the color indicating the actual TCR identity, we can see that there are large clones present, but that the identity of those clones changes over time. The color of the large clones shifts from time point to time point. Looking in the bottom left, we see both low clonality and stability. So here we see no dominant clones or no large slices of pie. And the color scheme is changing, indicating that the identities are changing over time. And then in the bottom right, we have high stability, indicated by the color scheme sort of staying consistent over time, but again, low clonality where there's no dominant clones. And in the top right, we have high clonality and stability, where we see those few dominant clones are staying consistent over the course of time. So when we overlay our data from the previous slide onto these potential states, we saw actually that we only observe the top right and the bottom left quadrants of this, where patients either have high clonality and stability or low clonality and low stability. And the responders tend to fall in the upper right again, and the non-responders in the bottom left. So from here, we're really interested in understanding, is there an underlying immune phenotype in these patients, say in the responders, that's supporting the ability of that TCR repertoire to gain that status of high clonality, as well as the ability to maintain those clones over time? So from here, we actually took the same 3 time points, the pre-treatment, pre-vaccine, and post-vaccine, and analyzed the same set of peripheral blood samples and used flow cytometry to do some more deep phenotyping of the B and T cell phenotypes that are circulating in the periphery of these patients. So before getting into the data here correlating back to the TCR repertoires, I just want to give a quick outline of the flow cytometry gating scheme that we use for these two populations. So for the T cells, it's a pretty simple gating scheme. We're gating single cells, gating out any debris in the second plot. Then we gate on only live cells, and we also have a gate here to gate out any B cells, and that's the CD19 gate. On the far right, we're gating on all CD3 positive cells. And then from there, we're bucketing our T cells into CD4 and CD8 T cells. And from each of these T cell subsets, we can look at the differentiation of effector, naive, central memory, and effector T cells by looking at CD45RA expression versus CD62 ligand. And we can also look at just generally memory versus naive effector T cells utilizing CD45RA and CD45RO. So those will be the main T cell populations that we'll be talking about today. And then for the B cell gating scheme, very similar here. We start gating out any debris. We're gating on single cells. And then on the far right, we're gating out any dead cells, as well as any other immune cell subtypes like T cells, monocytes, or NK cells. And from there, we go through a CD19 gate to hone in on our B cells specifically. And then we can differentiate between naive, non-class switched memory and class switched memory B cells by looking at CD27 versus IgD expression. So the naive cells are obviously going to be the least differentiated, moving up to the non-class switched as a sort of intermediate phenotype, and then the most differentiated being the class switched memory B cells. So when we actually take these gating strategies and isolate out particular immune cell phenotypes, we're able to correlate those with that cumulative frequency of consistent clones. So to orient on the top left plot, we're looking at the % positive effector memory CD8 T cells versus the abundance of that population G. So when we look specifically at the effector memory CD8s, we're seeing a positive correlation. So when a patient has a higher percentage of effector memory CD8s, they also show a more consistent clonal population in their TCR repertoire. And we see a converse correlation with the naive CD8s, where if a patient shows an increased number of naive CD8s, they have a lower clonal stability over time. And similarly, when we look in the CD4 compartment, we see the same effect. So a positive correlation between effector memory CD4s and that clonal, consistent clonal population, and a negative correlation with naive CD4s. So I think up until this point, it's probably not super surprising, the data that we're seeing here. It sort of makes sense that an effector memory phenotype in the T cell population for these patients would be supporting that ability to have a clonal stable clonal population over time. But interestingly, when we looked in the B cell compartment, we actually see a very similar phenotype there as well, where an increased population of class-switched memory B cells positively correlates with that clonal population. And again, conversely, the naive B cells negatively correlates with the clonal consistency. And we see in all of these plots, again, that the PFS9 patients are tending to sort of aggregate together in the right side of the quadrant. So for the effector populations, an increased population is going to correlate with the consistent clones, and all of the responders are falling in that category. So given this information, we were really interested in if there's an actual correlation between these T and B cell compartments. So on this plot here, we're actually plotting on the y-axis a ratio of class-switched memory B to naive B cells. And on the x-axis is a similar ratio, but comparing the memory CD8s to the naive CD8s. So a high ratio here is going to mean that the patient has more class-switched memory and more memory CD8 T cells. And that's essentially what we're seeing, is a positive correlation suggesting that the responder patients in green, who are skewing towards the top right corner of the plot, have both an increased class-switched memory B cell population as well as an increased memory CD8 T cell population. So a recap from this section, we've observed that the TCR repertoires of the responder patients are both more clonal and more stable, and that starts from the pre-treatment time point. And this repertoire feature correlates not only with T cell features, but also with B cell features. So we can hypothesize from here that the PFS9 responder patients are sort of coming from home ready for treatment, and that they have this immune state that's rendering them susceptible to the treatment, and that's allowing them to respond to the treatment. And this suggests that the non-responders are coming onto the treatment with an immune system that's unaware that there maybe even is a tumor, and they aren't able to elicit that immune response after being put on treatment as well. So this leads us to the final question that Asaf will go through next, which is, can we actually quantitate this immune state that we're observing using all of these peripheral measurements? So I'll hand it back to Asaf to finish up.
Thanks, Kristen. So we've presented with you today with all the measurements that we've taken from these leukapheresis taken in different time points, the TCR sequencing from which we derive Gini coefficient and unique TCR counts, and D50 and other things that we, in the interest of time, did not show today, as well as all the measurements coming from the flow cytometry, the extensive B cell panel, T cell panel, the subsets in these populations, and so on. And we've shown you pieces of signal that are indicative of response or non-response from each of these measurements. But what we really wanted to know and to be able to do is to take all that information together and kind of distill it into a score that we can use. And that score will focus on where the signal is coming from in all of the measurements. And because we want to be able to really make some sort of prediction, we've only focused here on time points, on data that came from the pre-treatment time point. So before any therapeutic intervention took place in this cohort. So what we've done is took this big matrix, basically, of patients by their different measurements, and we applied principal component analysis to this data set. And what this algorithm is helping us do, being a dimensionality reduction tool, is really trying to focus different measurements that contribute to the diversity in the data, the variance, into simpler scores that are fewer. So instead of looking across all of the different measurements, it's really trying to bring them down to the few key important ones, weighted together by their significance. So traditionally, we look at the first PCs where the most of the variance is explained. And when we look at PC1 and then at PC2, what we see in the positioning of our patients that while there's not a lot of diversity in terms of responders and non-responders along PC2, PC1 in itself is actually a very strong separator between PFS9 patients, which are in the green dots here, versus the no PFS9 patients. We tend to go with a more negative PC1 score. So we wanted to see, wait, so does PC1, the PC1's value, does that predict well for how well the patients are doing? So we first calculated PC1 versus the progression-free survival value, and we see that having a positive PC1 score indeed correlates with having a longer progression-free survival. So then our, of course, next and most burning question was, so what is in PC1? What's contributing to that? So we use a measurement or an estimate that's called the loadings, and that's part of the principal component analysis, where we can see which of these different measurements contribute the most to PC1. So you'll see here negative value and positive value. What this means is that, for example, at the very bottom here, having a positive value for effector memory CD4s will positively impact your PC1 score, or one sample's PC1 score. And having memory CD4s will have the same impact. And similarly, for having a high Gini coefficient, which, as you'll remember, is indicative of clonality, having effector memory CD8s, and so on. And conversely, having high values of naive effector CD4s or naive CD4 or D50, which indicates a non-clonal population, will reduce the score, the PC1 score of the sample. And what's nice to see here is that there's a distribution of the TCR measurements and the flow staining measurements that in the way they contribute to this PC1 score. So just to try to estimate the strength of the PC1 score, What we've done is arbitrarily chosen 0 as a cutoff and said, okay, so every patient that has a PC1 score higher than 0 will be in one group, and every patient that has a PC1 score that is negative will be in the other group. And then we plotted the Kaplan-Meier curve for the progression-free survival, and you'll see on top, these are the patients with a positive PC1, and here in red are the patients with a negative PC1. And this is clearly a very striking difference in their progression-free survival profiles. And when you quantify the median progression-free survival of patients with a negative PC1, the value was just below 7 months. Whereas when we look at the median PFS of patients with a PC1 that's higher than 0, for this analysis, the median has not been even reached after over 21.5 months. So to summarize everything that we've shown to you today, pre-treatment blood-based factors predict response to immunotherapy, and in this case, the immunotherapy was our personalized neoantigen vaccine, NeoPV01, plus nivolumab, the anti-PD-1 treatment. The TCR repertoire clonality and stability associate with improved clinical outcomes, and the baseline, so the pre-treatment T and B cell memory phenotypes associate with improved clinical outcomes. If we now combine this information, combine the baseline TCR repertoire and the PBMC phenotypes, we can predict the response in the trial to the treatment. And then these analyses suggest that there could be an underlying immune state that renders some patients more susceptible to treatment, which could help in patient stratification and medical decision-making. With that, we would like to first thank, our biggest thank you is to the patients and their family for their time and their pain and contributing the samples, which really enabled this study. We also want to thank the clinical investigators who treated the patients, who have collected these samples, and again, made the study possible. And to the broader BioNTech US team, this is, of course, involved many people if we're thinking about peptide production, the clinical efforts, bioinformatics, so really across the board. And of course, the iRepertoire team for their huge help in getting this data possible and helping us with creating the repertoire information here. Thank you very much, everyone, for attending as well.
Thank you guys for a great talk. So the first question we have from an audience member is, taking your results into account, would you infer that the neoantigen vaccines are not completely able to boost the immune system against neoantigens?
I can take this one. So I'll point you to the paper that we showed in one of the first slides from Ott et al. That definitely will outline that information in detail, but in general, yes, that's true. When we analyzed the immune response of these patients to each individual vaccinating peptide, we didn't see a response to every single peptide in every single patient. So I would say that there are particular antigen or neoantigens that are eliciting stronger responses than others. And that's obviously something that we're interested in tracking, and sort of feeding back into our recon predictor to understand if we can better format the peptide vaccine moving forward, targeting those neoantigens that did elicit sort of the strongest responses. And we've also done some detailed phenotyping beyond just, did they activate a T cell response? We've looked at, did they secrete particular cytokines? How did they perform in various immune-based assays to better understand those responses to each individual peptide?
So the next question is, do the expanded TCRs recognize the corresponding neoantigen or MHC?
Maybe I can take this question. So it's a complicated one. It's a great question. I won't have a super satisfying answer. When we look at TCR data, of course, just from the TCRs themselves, it's very hard for us to tell what it is that they're reacting to. Of course, we would need further sequencing and experimental validation. However, if you look at the paper that was published here, you will see that we did do some single cell analysis that pertain to specific neoantigens and their MHC restrictions, and we're able to confirm specificity. But I wouldn't venture into saying that all clonality in the study is necessarily driven by tumor antigens.
And have you checked for other immunophenotypes, such as exhausted states versus response or non-response?
Yeah, so I think in the again, some of it appears in the manuscript, and some of it was even in the breakdown of the PC1 score. There's also been PD1 staining on some of these cells, and there are other panels that have been incorporated into the analysis that we have not expanded on a lot today.
So another question coming into the chat, and I'll just quickly remind you guys, if you could drop your questions in the Q&A, that would be very helpful because then we can keep them logged in case additional follow-up is necessary. The next question is, how do you think the sequencing depth influences the association between the TCR measurements and the clinical phenotypes of interest?
Yeah, of course, a great question. We know that the depth is always a challenge in repertoire. Getting a good representation of the TCR repertoire, it's always a sampling problem. It's a sample of how many cells out of somebody's whole blood that we are able to collect, and then what happens in the library preparation, and what is the drop-off of different TCRs. To kind of avoid having these kind of issues, we try to maintain a very fixed depth per sample. So we always sequenced for 1 million reads per sample. And in kind of like QC analysis that we've done throughout the project, we also checked what happens if we perform subsampling analysis. So try to make sure that even computationally in silico, samples will receive the same sequencing depth. And the results have always been stable to those analyses.
So were any tests performed to observe similarities or differences between PSF9 and non-PSF9 populations, such as to identify what clones were beneficial to the responders that the non-responders didn't have?
I'll assume you're referring to the clonal similarity or overlap between patients. And we don't really see clones coming up across different patients. So we think these clones are actually highly private. They're not public clones that tend to occur across. And also, as would be expected from a personalized kind of treatment, these are different neoantigens. They're different from patient to patient. If we want to think about, can we identify features of a TCR sequence that are maybe beneficial? I think that's a very big open question for the field right now. Can you look at the TCR and say, what is it doing for us?
So did all of the patients in the study have similar levels of severity in their illnesses? And if not, maybe how did those sort of differences in severity impact the outcomes?
Yeah, I honestly can't answer that in too much depth. We do have a bit more of the sort of statistics on what these patients' disease looked like in that larger paper that I referenced at the beginning. I think overall, this is all metastatic melanoma for this cohort, right? Yeah, so these are pretty late stage patients, many of which have already undergone previous rounds of therapy. So I think, that's sort of the thing that is similar across all of these patients. Beyond that, I don't want to say anything incorrect about the statistics of the patients.
And did you do anything specifically to look at T cell effector functions, maybe differences between responders and non-responders?
Yeah, I think that kind of gets back to what Asaf was referencing, where we did do some pretty detailed phenotyping of the T cells in the periphery for these patients beyond just like the effector memory phenotyping that we showed today. And there are other phenotypes that correlate with response. So I think there's additional aspects that can be followed up on. And again, some of those are highlighted in I keep referring to the other paper, but there is a lot more information there that summarizes the study. And we did look at, both the CD4 and the CD8 compartment, as well as gamma delta T cells and these sorts of things to sort of exhaustively analyze what might correlate with response.
And did you look at the population of the antigens themselves within the patients to see if, there was similarities across all the patients, or if there were some sort of distinct antigens in different patient groups? Yeah,
I think pretty much across the board, there wasn't really overlap with the neoantigens that were present in this patient cohort. Asaf, correct me if I'm wrong, but I think there's very little, if any, overlap.
That is correct.
Was the immunogenicity of the tumors assessed in the patient pool at all like MHC1 expression?
Asaf, do you want to speak to the tumor?
Yeah, I'll have to check what we reported in. It's actually in another study where we focused more heavily on the tumor. There has been transcriptomic analysis, and I think, Kristen, correct me if I'm wrong, also IHC analysis of the tumor. But I will have to refer back to that paper to see what exactly was the finding there.
Another question, did you look at both alpha and beta chains?
Yeah, great question again, asked by a reviewer as well. So there's of course, that's something that is possible when we do TCR sequencing. In this particular study, we didn't have paired alpha and beta information, but we've sequenced the alpha TCR repertoire separately. And it was actually very striking to see how one-to-one the results were. So it did not matter at all if we relied our predictors and all of the PCA analysis on the alpha chain repertoire or the beta chain repertoire.
Did you look to see if the clonal TCR distribution was the same in naive central memory and the effector memory cell population?
Not sure I fully understand the question. I'll just mention, so since this is not single cell data in the sense that we can't get the TCR sequence from a cell and also tell what the phenotype of that particular cell, we're looking at the correlations across the bulk populations. But as Kristen has shown, there is a correlation across patients between having highly clonal populations and having more effector memory phenotypes of both CD8, CD4s, and even class switch B cells.
Are you guys planning to do any follow-up single cell work?
We've actually performed a bit of single cell work. It's been very challenging. We need to remember these are patient samples, but we have, I think, at least 8 reported in this particular study that you can look up. It's one of the supplementary figures. It's where we have done confirmation through a single cell perspective. But again, even in that analysis, we only have the sequencing measurement from those cells and not phenotyping of the particular cell.
So one audience member asked, if the nature of the high frequency clones aren't changing in the responders over time, and they're not shared across patients, could the higher survival rate be attributed to the response to immunotherapy or a higher innate resilience against cancer?
Yeah, I think that's sort of the point we were getting at in the middle, suggesting that when we see these patients having a clonal stability over time, that's also supported by that effector memory phenotype. And that's happening, we're seeing that at pre-treatment, right? So that's happening before the patient even comes in to the study. So that would suggest that they do have this sort of, I guess, as the questioner is saying, an innate resilience against cancer, where their immune system is heightened, it's alerted to the cancer, compared to the non-responders, which just don't seem to have that sort of immune response that's gotten started before they're coming onto the therapy. So that's exactly right. That's what we're theorizing.
And if it is just an innate resilience, still being able to identify that innate resilience would be useful clinically, right?
Absolutely, yeah. We don't want to put somebody on our trial that isn't going to respond, right? So if we can do a baseline phenotyping quickly of these patients and say, oh, they have an immune response that's likely to respond to this treatment, they're an ideal candidate to come on to a downstream trial like this and hopefully succeed and respond.
Did you see any impact in your repertoire data based on the age of the patient?
So it is a well-known fact that age corresponds to clonality, and there tends to be a higher clonality in more aged individuals. In this particular study, the age groups between the PFS9 and the no PFS9 actually had no known difference between them. The only small discrepancy was with the healthy donor population that tended to be younger. We've performed different regression analyses to try to address this, but the result remains significant regardless. So we weren't concerned about that.
Excellent. And just one last question. What are the next steps for this work?
Kristen, do you want to go?
Sure. So we did have 2 additional peptide vaccine trials, NT002 and 3. This was when we were Neon Therapeutics before becoming part of BioNTech. And I think as sort of the world knows, we're shifting now towards more of an mRNA-based platform for vaccines. So we're taking the technology and the information that we learned from these peptide vaccine trials and applying that obviously to ongoing trials that are being designed with the mRNA vaccines. But overall, I think that's the progression of where this treatment modality is headed. And we can obviously utilize the data from all these trials collectively to identify patients that are likely to respond, potentially design new combinations that can enhance the response to the non-responders, et cetera.
Excellent. Well, thank you guys so much for a great talk and a really great Q&A. Audience, thank you for all of your questions. If you have any additional questions that pop up after the fact, you can feel free to reach out at info@iRepertoire, and we'll be happy to answer any questions you have more specifically about the technology or the services that are offered by iRepertoire. I'd also like you all to stay tuned for more webinars to come from iRepertoire. Just keep an eye on your inbox and we'll let you know when the next event is coming. And a big thank you again to Kristen and Asaf and BioNTech. Thank you guys for coming and sharing your great work with us all. And thank all of you for attending. Everybody take care and have a great day.